right, well, you go.
Hello, yeah. Okay, hi, good morning, everyone. Welcome again to the December version of the ALT2 meeting. This is our last meeting for the year. Just to remind everyone, the meeting is recorded to the cloud, so just be careful. Yeah, so today we have a number of presentations. I'm going to give an overview, some of the status update for the program. I don't know if Michelle, are you online? Yeah, we were going to have Michelle from NADA's lab give us an update of some of the in vitro and in vivo models that they're trying to develop. Apparently she's not online yet. Michelle? Yeah, I guess she's not online yet, so I guess we're gonna get started.
I'm gonna cut. Yeah, this is Nana. I'm gonna text Michelle and see where she is. I'm at a different location, but I'll see where she is. If you don't mind starting with something else.
There's a lot of echoing. Who was that? Hello?
Hello, can you hear me?
Yes. Who is this? This is Nada or Michelle? Michelle?
This is Nada. I'm at another location. Unfortunately, I mean, that's how I.
Okay. Yeah, there's a lot of echoing. I don't know if it's the best way to address that.
We can't hear you well, Nada. Yeah.
Hello? Oops.
She'll probably try to call back in.
Yeah. Okay. All right, yeah, we'll give another chance to try again. Okay, so with regards to the agenda today, like as I said, I'll give a quick overview, some of the status updates. Michelle from NADA's group, like as I say, is going to give us an overview of some of the models that they're trying to develop. Then we're going to get an in vitro assay update from Alex and Jean-Fru's team. Sue Cramp is going to give us an update on some of the efforts on the chemistry front, and Lisa has a nice little update on some of the in silico analysis for some of the ALK2, ALK6 selectivity that she has been working on. And then we'll have a lot of time to discuss some strategy at the end. So I wanted to give the group a bit of an update. On November 19th, the grant that we were on that was driving this project, we had to give an update to a committee called the TPAC committee, and for the benefit of others who probably didn't know when the program started, this grant was offered in December of 2017, and it was a 2-year grant. So we actually had a couple of timelines and deliverables. So what I thought I'll do here is just give a nice executive summary of some of the accomplishments. I think the team has done an amazing job in terms of really digging into the role of ALK2 in a disease area of real unmet need. We know the genetics of ALK2 is one of the underlying causes, but there are many other factors that contribute to it. But we think there's an opportunity to really go after this disease in a targeted fashion. So we think we've actually followed up with some genetic knockdowns and looking at how ALK2 inhibition may play a role in the proliferation of some of these cells. We have identified small molecules that are now CNS penetrant. Previously, a lot of the ALK2 inhibitors were not very CNS penetrant, typical of many kinase inhibitors. So I think we have now developed some nice CNS penetrant inhibitors that might have promise going forward. We were able to implement a very robust screening cascade to enable compound development preclinically, and we have identified very potent, selective, CNS penetrant and orally active compounds ready for proof of principle. Right now we have about 5 lead candidates, and it's kind of hard to pick, but that's going to get whittled down soon as we have a strategy going forward in the next 12 to 18 months of how to proceed with that selection process. It's important to highlight to the team that we also identified ways of mitigating potential liabilities. So that was good. And I think one of the challenging areas for a project like this is that there was no good, models, and so we've actually expended quite a bit of effort understanding some of these cell lines, trying to get a better handle on which ones are dependent on ALK2 for their growth. And, we're really happy to have groups like Angel and Nada and Chris Jones, who have actually had experience, this clinician who have had experience with some of the patient-derived models to really enable some of the in vivo model and efficacy development. So, I think, kudos to the team for really doing a great job of bringing the program this far. Like I said, we had a meeting with the TIPAC, and one of the requests at the last meeting, I think Alex had proposed whether or not we can get an extension, and I was happy to report the TIPAC has granted that extension. We had asked up until March, but they gave us up until June 2020, so that gave us a lot of time to really accomplish any little end games we might want to kind of finish up for the project. And so to accompany that, I just thought I'll put up some of the final deliverables that I had actually mentioned the last meeting that we were trying to accomplish. One of them was to at least complete the efficacy study with Angel and one of the molecules from the program in the xenograft burden model. So that experiment was done. We did complete it. We did have some concerns about the confounding effects of the vehicle. So on the right side, we're asking a couple of questions. Should we repeat with the new vehicle the same compound? Or do we test a new compound in the new vehicle? So those are things we're actually going to discuss going forward. Another actionable that we had for deliverable was demonstrating compound-dependent modulation of a PD marker, and we sort of selected hepciding as a viable path forward. It was a new venture. So what we had decided to do is run a pilot experiment, which was done by Charles River. That experiment is complete. I'm going to show you the data, but the data wasn't extremely positive in terms of the kind of robust response we were hoping for, but it'd be interesting to see what members of the team have to say. So the other question is, for the next 6 months that we have, should we try and find another marker or try and find an alternative route, like looking at a labeled compound and looking at occupancy in the pond as a potential route forward in terms of target engagement. So those are just some of the milestones that we had promised. So we feel like we have to go back, do the efficacy testing with the new vehicle, and whether we do it in a different model too. So those are just some of the things we're going to come back to the end. In terms of some of the action plans that we had last month, I want to give an update of where we are today. So we talked about scaling up the 5 lead compounds, but I did mention to you that 2 of the compounds were more fast-tracked because everything was in hand, and 2 of the other compounds were delayed because we were waiting on a material that wouldn't be delivered until, I think, in January. So that's ongoing. So we're hoping by the end of this month, M4K 2117 and 2163 hopefully should be delivered. So keep our fingers crossed. Last month we were waiting on some data for M4K 2163. That's one of our most potent in vitro compound. We got the rat picker data. We were waiting on the mouse escalation data in terms of determining whether or not we're going to see nice linearity with dose, and that data just came in, so I'll give you an update on that. We continue to kind of look at models. So Angel have completed the experiment, our first efficacy experiment with small molecule of the team. Like I say, we are debating whether we repeat that experiment with the new vehicle that we've identified. We also have Chris Jones and Deanna, they're also looking at another model. So that actually is in the process. I think it's ongoing or it's about started. And now, interestingly, we're going to get some update from Nada. Nada is developing some new models with some interesting outcomes, particularly with regards to ACVR1 knockout in some of these parental lines. So we look forward to that update. That could be a very interesting model to consider going forward. And as we continue to kind of dig into some of the biology of ALK2 within the disease, we're trying to develop tools. So there's some of the PROTACs that we're trying to develop. Groups continue to do CRISPR to understand some of the ALK2 dependency of some of these cell lines. The Charles River team continue to work on you know, the backup compounds, the blueprint backup, and also designing PROTACs that might potentially be CNS penetrant. So we got some more time to accomplish some of that. Again, I'm going to give you an update on the turpentine model. That was an action from last month, and so it's now complete. And, you know, we continue to explore, you know, the roles of ALTA in addition to other mechanisms that's driving the disease. And be nice, Michel Manger's group just published a nice paper on some efforts on looking at combination studies, and some of it aligns with some of the findings we've had so far. So I'm actually going to speak to a little bit of that. But the key is that we had requested a no-cost extension for the program, and I think I'm happy to report to the team that the CTIP the CTIP advisory board did give us a no-cost extension up until June, so we have a lot of time to accomplish some of the loose ends. With regards to some of the data update, like I mentioned to you, we had gotten some rat data for 2163, and the data was amazing, getting very, very good oral exposure in both rodents models, that is mouse and rat, as you can see. What's more interesting, like as I mentioned to you, we did a dose escalation study just to get a better sense of whether or not we're going to get increase in exposure with increasing dose, because this is essential when you start looking at tox. That's going to be an essential area in terms of compound selection. Can we actually dose escalate the corresponding increase in exposure and actually start to get a better assessment of the tox for these lead molecules? And so we're quite happy to see we were getting very, very nice linear exposure with dose. I mean, the bottom part of it.
I'm sorry to interrupt, the slide is cut. How about brain penetrance? I see brain at 2 hours and brain/plasma, but I can't see any value down.
Pardon?
You can see the bottom part.
Yes, yeah, we have the
same problem, yeah.
Can you just shrink it down?
Yeah.
Okay, let me do 6. Go to, yeah. So you can see that now?
Yeah, I can see it better now. Yeah, I can see it.
Yeah. So what's nice, as I say, this is one of the compounds that so far we've seen very good efficacy in the in vitro cell lines. What's even nice, so we're getting nice linear exposure with dose. And one of the things we did during this experiment at the 100 mg dose, since if that's a dose we are thinking of doing a lot of efficacy with, one of the things we decided to do is just get a snapshot of the brain exposure. So we took this compound and we actually looked at the brain exposure at 4 hours, brain and plasma, and we're getting very, very good brain penetration with this. And the absolute brain concentration is very, very good. We are seeing up to close to like, you know, what's that? Almost 30 micromolar exposure with the compound in the brain, dose at 100 mg per kg. So I mean, if this compound is active in the cell lines, I expect to get nice efficacy with this in the model. The other thing to keep in mind, again, like as I say, in the table, we had one from 10 mg up to 100 mg. You could see the Cmax, we're getting nice and, you know, linear exposure with dose. What's also important is the area under the cursor over the dose give you a good sense of linearity. That's supposed to be on an average, a nice little constant number. And so that's looking really, really good. So this seems like a very promising compound also. Can't wait to see this compound.
What's happening with half-life there at 100?
That's, you know,
Because you usually get the true half-life from the IV arm. But in this study, we just do the oral dosing. This is oral dosing. So as you start going to higher doses, you start to get saturated exposure. So sometimes the compound just stays so much of it in the gut. It kind of just leaches in, so you get this kind of delayed half-life. That's why you don't use oral dosing to determine the half-life of a drug, because you're going to get this variability depending on the dose you use. Yeah. But you just capture that element anyway. You know, it's just one of the parameters they collect when they're collecting data. Okay, but I think 2163 is a very interesting looking compound also going forward.
So going back for a second, interestingly, the thing about, this is 2163, that's
Yeah.
about 2009, because we were dosing that at 100 mg per kg.
Yeah.
And we would have had all sorts of inefficiency in that dosing level if that's because of the half-life, because it looks like it's staying in the gut, it's not being absorbed.
No, the half-life for this, if you look at, so the actual half-life we get from the IV ham. So here is the actual, so 2163 is about 2 hours. It's almost the same in mouse as it is in for 2009. Yeah, so the second. And then in rat, it gets better. It's about 4 hours. And that's not unusual. I mean, for mouse half-life, the question is how much. This is typical of typical compounds in mouse. Sometimes some of them have a half-life of 5-6 hours, whether or not that's what you want. But this is not unusual. It's like when your half-life is like you know, like 30 minutes and stuff, then you kind of get overly worried. But with the high turnover in mouse just metabolize things really fast. So 2 hours is decent. I mean, if you could dose twice daily, it's actually not too bad. And it's getting better in rats, and we're hoping it's going to be even better in non-rodent species. We expect dogs and stuff. Yeah. So it's not outside of what's typical of a lot of compounds that we've seen. So I'm not losing too much sleep over that.
Sorry, do you have any, do you have protein binding or brain tissue binding for this compound?
Yes, so this one was one of the most highly protein bound of the 5. It's about 99% protein bound, but the other lead compounds actually have a lot more diffraction than that. Yeah. Well, that's a good point.
And that's bound in the brain.
No, not brain. This is not brain binding. This is just plasma. Yeah, we have done. Yeah.
So you don't have brain tissue binding?
No, we don't have brain tissue binding for that particular compound.
Okay.
Okay. So onto the hepcidin data. So again, for the team, one of the things we're trying to do is to see if we can identify a nice and convenient PD marker for ALK2 biology, something that we can dose, take plasma, and get a good read of ALK2 target engagement in vivo. And so based on some data from a patent that Sue had sent to us from Novartis, we thought this was a good model to really do a little pilot study with. So we took on the task of having Charles go look at this model. It's a very new model to them. And the idea was to use terpentine oil. When you inject terpentine oil sub-Q, it's supposed to induce hepcidin production in blood. And then that you can measure. And the hope was that process appeared to have some ALK2 and ALK3 dependency, as Alex keeps reminding us. So, based on the Novartis paper, when they dose terpentine oil, they saw an induction of about 40 to 80 nanograms. But in this experiment, when we dose terpentine oil, we only seen about 1 to 2 nanograms. And so that was very, very low in rats. So we don't know for whatever reason why that is. However, after the induction, if you dose with doses of the compound and you check 6 and 24 hours later, if the compound is working, you see this nice decrease in that induced hepcidin from the terpentine oil, so you would expect a nice reduction. But here we never saw any very significant, there's a slight reduction at the 24-hour time point, but it's not very significant. So, and as I said, this was a little pilot. If the data was robust, then we would have subjected the other lead compounds to it. But based on this data set, we're questioning whether it's even worth pursuing with the other compounds. And so it's up to the team, but I think, for me, I think this is a no-go decision in terms of using hepcidin, and we might want to start looking at alternative markers. The other thing I'm thinking about, like I mentioned earlier, I think that's going to have a lot of legs going forward. For a lot of the lead compounds that we have groups that we can actually PET label. And so I think one of the nice studies in a disease like DIPG is to just find out how much of the compound is getting to the actual pun where the disease arises. And so if you could label one of these, and you can actually, in a phase 1, do like dose escalation and see how much of the compound is actually getting to the pun. You get a good sense of, is the compound actually getting to the target? Because that's one of the problems a lot of times. A lot of these compounds, they may get into the brain, but apparently the pun's barrier is a lot tighter than the total brain BBB. So it's good to be able to determine whether the compound is actually getting to the target. And so something to think about going forward is some of the lead compounds we have, like compounds like 2117, which have an N-methylpiperidine. Those are things you can label with PET. You can label that methyl group with PET late in the process and do a quick PET study. Compound gets into the brain, gets to the target. You can do dose escalation to see when the pun is fully occupied with compound to sort of gauge your phase 1 doses and stuff. So those are things we might want to discuss going forward.
Before you leave the slide, does anyone think we should do the hepcidin study again? I don't see any compelling reason to do it, looking at the error bars, et cetera, and what we have. It was a good attempt. It was a smart thing to try,
yes,
et cetera, but I don't think the results were what we were going to do.
Only comment I would have is why the induction is so low. There is a huge mismatch
Yeah.
from the literature
Yeah.
and what we have here.
Mm-hmm.
It's like, 20 times less than what we were expecting. So,
Yeah, they followed, I mean, they did follow the Novartis protocol that was outlined in the patent. So, and this is what we got. We didn't get that robust
Yeah.
response.
No, considering where we are and the resources we have, I think we've passed. Nothing. Okay, thanks. We can move on. Let's just note that on the file.
Owen.
Yeah.
Can I make a comment? I'm not sure why I'm echoing.
you have too many.
Go ahead.
Okay. We're still echoing. Hang on. What about now?
Yeah, that's much
Yes,
better.
that's better.
So, looks like you're the Pink Floyd band.
There's a strain of mouse that's bred to have a BMP response element linked to LAC-Z, beta-galactosidase. So you have an indicator mouse. And so you can presumably dose that mouse and see across the whole mouse in every tissue where your BMP response is going up or down or regulated by the drug. I think I know a colleague in Oxford, I think, has one in locally, but I presume they're worldwide, probably commercially available, or probably, I don't know, probably a few people have them, but there's some literature on it, so I'll share that with you, and we can look at it and see whether it's relevant for the pons or the brain.
Okay. Okay, it's a good idea.
Thanks.
All right. Thanks, Alex. Okay, so just wanted to remind the team, so these are the 5 lead compounds that we're looking at currently. Should I go back to full screen? And so one of the questions we keep asking ourselves, I mean, we have this dashboard of lead compounds. It's a lot of compounds to put into development, but we don't know what's actually going to rise above in terms of, properties for each of these compounds as we put them into some of the more advanced studies, whether they're dog telemetry or just simple rat tox. So we don't want to kind of prematurely throw out any of these compounds, but eventually this list has to be whittled down. And so the bigger question was, do you just focus on the CNS penetrant ones, that's the top 3, or do you pick 2 of the CNS penetrant ones and take a risk on one of the less CNS penetrant one with, the decreased herd risk? I mean, so those are some of the kind of questions we're toying around with. Because over the next 6 months, I'm hoping that we can at least get 1 or 2 more compounds in an efficacy model. I think that's realistic. I don't think we can run all 5 in an efficacy unless somebody actually has the manpower and stuff and the resources to do that. So, we're starting to think long and hard about, okay, if we're going to pick 2 or 3 of these compounds to really get some data by June, what should be the selection process, charging the selection process. So that's some food for thought. I mean, so we're debating, do we do the all CNS penetrants, which are likely to get into the brain, or we pick, we've actually looked at 209, 2163 is one of the most potent in vitro compound that you've seen today. Do we go with 217, 2163, and then pick one of the AMIs, which is less CNS penetrant, but of lower herd risk? In terms of when the compounds are going to get delivered is another factor. We had scale up 2009. We're hoping by the end of Christmas we'll have the other 2. So just by practicality, the CNS penetrant compounds might be the first ones that are available to do any large efficacy study. So just by default, there might be the 2 compounds that we might have a chance to evaluate. Sorry?
Alex,
yeah?
Alex again. Do we have a compound which is the hybrid of 2117 and 2163, i.e. 2117 but with the fluorine?
2117.
Yeah, instead of pyridine, you have
we did the NH compound of that one, but we don't have the N-methyl.
I don't think so, no.
As a matter of fact, we're actually looking, the student who was working with Ahmed, Mehak, who presented the last time, she's actually looking at some of the old compounds because she's trying to develop this MDCK model and kind of trying to predict. We picked one of the compound with a fluorine there, and it looks decent in terms of its CNS profile. I think relative, I don't think we've done full PK of it yet, but
Yeah,
but we need to evaluate the PK profile of it and stuff. I mean, one of the things we were also cognizant of, when you have halogens there, because we had done chlorine and fluorine, and then we were also concerned about that position can be very labile for reactivity in a pyridine. So unless the compound actually has stellar properties and stuff, that's why we like the methyl. I mean, it's not as reactive as a center. But that's a good point. We could go back. We'll see. I made it looking at the fluoro compound. If the PK look decent and it's compelling, we could go with it.
So now we're up to 6. We're going the wrong way.
What's that? Oh, now that's a new compound. We have to get a lot of data on it. I mean,
No, I was just wondering if the methyl in the solvent channel part changed the HERG from the 2,500, whether that would increase it.
oh, this. We had. I remember we had the cyano compound was just as bad. So I mean, we had put cyanochlorofluoro. We had sent the cyano. The cyano was a good compound. The cyano had very similar HERG values to those. And the problem was then in CYP. So, the cyano was a strong compound looking good throughout the board, and then bam, we saw the CYP data and we realized it's a very potent CYP inhibitor. So, I mean, whatever group atom we put there, I mean, we still have to do the full profiling and see if it actually has any additional liability. So, yeah, I mean, it's funny, I mean, because, we're looking at the HERG, we've done SAD to mitigate the HERG, and from what we have learned, as I say, I mean, the pyrazine SAD didn't fix it, unless you're in the properties of the molecule. We can fix HERG by, like as I say, getting rid of the basicity, like we saw earlier, but when we do that, the molecule becomes very insoluble, and we lose all that nice PK properties. So, I mean, as I say, based on what I've seen with the HERG, I feel like it's a risk we can take going forward. We should do dog telemetry. I think that's going to be the final decision maker for triaging these compounds. I mean, if these HERG numbers was like, 100 nanomolar, 200, then I would be overly concerned. But in a disease area of really, really unmet need.
One possibility would be we could do the same thing we do with another program, is just look at the sodium channels.
Yeah, we could do the profiling across all of the other channels.
Yeah.
Yes.
Just do an in vitro
That's a good idea.
assay.
You heard that?
So this was something we discussed with my CRL colleague a couple of months ago, wasn't it?
Exactly.
They can certainly do some of these assays to look at the profile on the other channels.
Mm-hmm.
The other question I had is, just following up on what Sue was asking, is it easy to measure that brain tissue binding, and would that be a criteria that we could use ahead of efficacy?
So we did 209, we did one compound, and I mean the brain binding data was kind of high, but the question, it's not cheap. That one is a little bit more expensive.
Again, it's something we can do at CRL if you would like it done.
But my question is about the brain binding isn't necessarily a bad thing. Like, I mean, you're not getting a lot of free fractionation, but you might be extending the half-life inside the brain in that area. I mean, I guess we just don't know. I mean, there's just so many other pieces to this, but it might not necessarily be a bad thing.
No, it might not be. I mean, and it looks like you're getting a nice boost in half-life with high plasma binding. So, and it's also interesting just to ponder whether or not the plasma binding and the brain binding would be correlated.
Mm-hmm.
So my thought on that one is
probably not necessary to.
Yeah, I wouldn't be sure I could pursue it if we don't know whether there's going to be a benefit.
If it doesn't give us a clear answer.
Yeah, I think doing the ion channel for the HERG gives us a peek at it. I tend to agree, Matthew, with you just for whatever it's worth, and I'm not sure that it is the critical tox problem with it. The problem I have is I don't think we know enough about the PK/PD at this time to really knock any one of these out. And that that's my thing. I mean, we don't know how much drug we need to treat this. We don't know, what's the dosing level. We don't know anything about a lot of this stuff. And to start kicking them out now when we really don't know, like, what's the driver? Is it AUC? Is it time above? Is it, you know, what are we looking at? And each one of these might have very different characteristics for what's actually making the drug efficacious. So as much as I would love to get this down to 1 or 2, I'm not sure we know enough. I just don't know.
And I know
someone else can come up with something. I'm all ears.
Well, I know you've shown the data in the big tables in the past, but is the cellular data comparable to the enzyme data? I mean, in terms of the rank order and all that?
So that's the question. So the cellular data that we have, I mean, so we were trying to pick a dependent line, which we thought was the DIPG21, appeared to be a line that seems to have dependence on AL2. That still needs to be absolutely borne out with a lot of genetic biology.
I guess I just meant the PSMAD, the functional cellular data.
Oh yeah, so we have only put 2009 in this PSMAD. I think you did, Jerome had done that experiment. We could probably test, I mean, Jerome was kind of tied up for the past couple of months, so now we have the 5 leads. That's the other question, Jerome, whether or not we put all of these in the phosphosMAD and the EAD1 assay just to see how the cellular functional activity is. That could be something to help rank order some of these compounds in terms of priority. So
just another comment there as well is to think about DogPK at some point. So if you go to dog telemetry, you're going to need to have it anyway, but it might be a nice way of triaging the compounds.
That was kind of my hope. I think the best way, like Owen always talk about, he wants to see more tox. That could be a kind of a way to triage. It could be toxicity, and it could be the dog telemetry. I mean, we put them all in dog telemetry, and you realize 1 or 2 of them actually doesn't have that.
But I was talking about just an IV PK study in the dog or PK in the dog. So because once you've got 2 species PK, you can also start working out dose calculations as well. So that might help to triage.
Exactly. I would love us to get some dog PK. I mean, that was a costing. We were determining whether we do that after the CTIP grant. So that's something Owen is thinking a lot about. But that could also help.
Right now, it's something we have put forward as the next grant, etc. So I think someone just tried to raise their hand or something. Maybe there was a flash up there. I think it was. I'm not sure.
Somebody?
I'm sorry. Hello? It's me, Nada. I'm sorry. I know that Michelle had a problems setting up the audio. Now she's set up. Just I need to leave in 20 minutes, and I would like to listen to her presentation and kind of address things. If it's possible to beg the favor, even though we're late, and I apologize for that. She presents.
Okay.
So
okay, let's go then. Let's go to your presentation, Come back. No problem. So I'll switch, how do we, so you're going to take control?
Hit escape first.
So,
just shrink down your thing.
So, Michelle, are you there?
Yes, I'm there. So I will just have to share my screen with you people. Okay. So
Stop sharing.
Speak up. What we've been doing is that we had a few sublines that we got from different people and some that we generated on our own, where we CRISPRed the ALK mutation. Either we repaired it in some of them or we removed all of the ALK. So we wanted to create a to interrogate and.
Okay, sounds good. Okay, so you have control, Michelle?
Okay, guys, so you're able to see my screen?
Yeah.
So I'll be presenting here just a brief update on the work we're doing on some in vivo models of the DIPG as well as in vitro models, mainly on screening different classes of ACVR1 inhibitors in the IPG. So just very briefly, just guys for you, I know you know, but it's important to just tell you what we're working on. So our lab is interested in studying pediatric glioblastoma, focusing mainly on the epigenetic modifications that underlie this disease. This is mainly because there are multiple histone mutations that were identified in pediatric GBM And
Tell me, you know very well the disease. Skip this slide.
I'm sorry.
Skip the slide.
Just for you to know that we are focusing on the IPGs, especially on the ACVR1 mutation that is present in 30% of the IPGs. What is very interesting for us is that there is a wide spectrum of ACVR1 mutations that underlie the IPGs, unlike FOP, and we're trying to decipher how this differ between those H3.3 and H3.1K27M. So in order to understand Vestis disease, we hypothesized that ACVR1 potentiates K27M into ureagenesis and is actually might be a therapeutic target. So we underwent a CRISPR strategy whereby we tried to knock out ACVR1 using the strategy that we have. We were able to get 3 different types of clones. One where we will have a frame shift only in the mutated allele, leaving the wild-type allele intact. And here we did it only for the R206H mutant, the IPG. And one where we will have a frame shift in both alleles. So here the complete gene is knocked out, and we will have the ACVR1 knockout. So there are different cell lines that we worked on. We have 2 R206H ACVR1 mutant, the IPG7 and PT869, in which we will have complete knockout as well as R206H knockout. And we have 2 H3.1K27M ACVR1 knockout, the IPG4 and 36, where we obtain only a complete ACVR1 knockout. In order to test the importance of ACVR1 mutation in the IPGs, so we tried in vivo models in which we injected into pons the IPG7 and BT869, whether it is the parental cells that are not not edited, or the knockout cell lines. And as you can see, in both cell lines, knocking out ACVR1 increased survival of the mice. So this is really significant, and even in BT869, the ACVR1 knockouts are still ongoing. So this was very interesting and very important too. We tested the same in vivo model, but now in sub-Q models for the IPG4 and the IPG36. And also, as you can see, our knockout cell lines had more increased survival, and also with less tumor volume evolution, as you could see in the IPG4 and the IPG36. So that was, it is very interesting for us. So we know that ACVR1 could be a therapeutic target, and as you all know, there are lots of multiple compounds for ACVR1 as ACVR1 inhibitors. Lots of them are non-specific and have off-target effects, as you can see in this panel. That it's a very popular and that I've used, and only type 2 of them were more potent than others, but they also have a wide effect on other wild-type or ACVR1 wild-type cell lines. So there are 2 more compounds that were a bit more specific, so especially M4K 2009, that is a very specific ALK2 inhibitor, and I've also used the TP0184 that was in clinical trial for the IPG, not so beneficial, but anyway, I've tried it on multiple cell lines, and they also showed a wide range of efficacy on these cell lines. And very interestingly, this TP0184 showed, the cells were more vulnerable to it, maybe because it is not a very specific inhibitor. And just to check if our drugs are specific for ALK2, what I did is that I used these 2 drugs in ACVR1 knockout cell lines. So I'm showing examples of the IPG4 with the IPG4 knockout ACVR1 and the IPG7. So just focus on the M4K 2009. As you can see, when I knock out ACVR1, you have much less sensitivity of the drug towards these cell lines compared to the parental cell lines. But also very interestingly is the IPG7 cell line. Just because we have those R206H knockout, in which, remember, we have one wild type allele that is still intact. And as you can see, these R206H knockouts still respond to the drug. However, in the complete knockout, the cells are irresponsive. So that's very interesting, and this did not happen for the DP0184 drug, meaning that ALK2 is, M4K2009 is really specific for ALK2. So we've shown that, we and others actually, we've shown that K27M cell lines are actually vulnerable to EZH2 inhibitors, and it was very important for us to see whether we could combine ALK2 inhibitors together with EZH2 inhibitors. And for this reason, I did a combination experiments, and really what we could see is that there is actually a beneficial effects when you combine the two drugs together in different cell lines that I tested. So this is a summary of all these, of all the ranges, and this is very important because these cells that are actually K27M and ACVR1 mutated cells, they have the range of synergism is really low compared to the cells that are either wild type for ACVR1 or also wild type for K27M and ACVR1. That is really interesting. I've done these studies with other ALK2 inhibitors other than M4K2009. We also find a very interesting synergism. So just a small summary of what I've shown is that ACVR1 actually potentiates the effect of K27M in vivo and is a good target for treatment. Among the multiple classes of ACVR1 inhibitors that were used, M4K is very specific for ALK2 inhibitor and doesn't really, the knockouts do not respond to the drug. This is very interesting. And combination of ACVR1 inhibitors and EZH2 inhibitors are very beneficial and can give a synergistic effect. But also it's very interesting to mention that other epigenetic regulators can also be used together with ACVR1, because I've used with HTAC inhibitors or HAS inhibitors, we have a very interesting synergistic effect. So for our future directions, it's what I'll be doing is that trying to use the M4K2009 in vivo in sub-Q and orthotopic models of our models that we have, and also other to test other compounds in the IPGs. So that's the plan for our future directions. And that's it for me. Thank you.
All right, thanks, Michelle. That's very nice update. I mean, some very interesting data. I mean, a couple of questions. So the EZH2, I mean, we have tested some EZH2 inhibitors in combination, and we didn't really see much synergism with them. So it was interesting that you're seeing synergism with the EZH2 inhibitor. We had tested the clinical one, tazemetostat. They are very similar, but one of the things we knew was an Achilles heel for a lot of these EZH2 inhibitors is that they're poorly CNS penetrant by virtue of a pharmacophore intrinsic for the EZH2 biology. So what we had done is the EED inhibitors, which is supposed to phenocopy a lot of the EZH2 biology. We had tested the EED inhibitors, which are a lot more permeable and more CNS penetrant. And also we didn't see any synergism with those. So it would be interesting for you to actually explore the EED compounds.
Okay.
It's supposed to phenocopy the EZH2 biology. See whether or not you actually see that synergism, because those have better chance of penetrating the brain. And I mean, we have a number. There's one clinical EED inhibitor from Novartis. We have it in-house. So I mean, if you really need some of those EED inhibitors to further validate the EZH2 combination with ACVR1, yeah, we can actually provide some of those compounds to you.
Yeah, sure.
So when you tested those EZH2 inhibitors, did you test them on the same cell line that you had?
On the same
or on another one?
Yep, so we did 007.
We did 007, 36, 4, 21.
Yeah, PT21, yeah.
I
Yeah,
have
we did.
a comment for the EZH2 inhibitors. So we have a few of those EZH2 inhibitors for in vitro studies. Not all of them were really potent on our cell lines. So I found this UNC 199 being the most potent. I know it's not brain penetrant, but I mean, I've used it in vitro. For the EED inhibitor, we've tested one EED inhibitor, which is the A395, yes, but it was pretty high, very high range, like very high concentrations to, at least in vitro. So I haven't tested others. So if others are more potent, I will be very happy to test them.
Yeah. Okay, yeah, we can provide those.
Hi, this is Peter Howe. Sorry, I'm joining the call for the first time, so great to meet you all virtually. Tazemetostat, certainly a drug I know a little bit about, and so there's never been any good evidence that it is CNS penetrant. Certainly, I understand that you'd be looking at EZH2 combinations, in a sort of test system now, but that one's probably one that doesn't have legs for what you'd ultimately want to do here. Certainly, if one can develop a brain-penetrant EZH2 inhibitor, that could be very interesting. The other caveat I would say for EZH2 inhibitors in general, but specifically, but epigenetic, the current class of histone methyltransferase inhibitors is that really the activity takes a while to develop, and so typically even in vitro models, a much longer duration of exposure than even 3 to 5 days is generally necessary to see a phenotypic effect. In the past, others have used 7 to 14-day exposures just because.
So
of the
this is exactly.
effect.
I completely agree. So this is Nada Jamato. So again, this is also my first time joining this call. I agree completely with you. Anything that has to do with the epigenome and any remodeling takes more time than the regular killing and whatever. I think we could even more state that maybe the cell death is not also one point that we should be looking at, but more cellular differentiation, because this is part of the thing. You're reshuffling maybe to something that can differentiate better. The reason to use EZH2 inhibitors is based on the biology that was presented by Christian Hellen's group and by us, because we think that this is the residual piece in the K27M mutant that our K27ME3 might be kind of maintaining cells. And we wanted to see if, or at least in vitro, we could have an effect with EZH2 inhibition, further EZH2 inhibition. So again,
absolutely.
Other studies, this is so after that you can kind of go and see if there's anyone that's CNS penetrant as they come.
Right. Nothing would make me happier to see EZH2 activity in a pediatric tumor. The one thing I would add to your comment is that in the past, what we found when I was previously working on this is that when groups presented short-term exposures of EZH2 inhibitors and found cytotoxicity, within the 72-hour time frame, it generally related to off-target effects. So, in agreement with what you just said.
Mm-hmm. I completely agree with that. So we have, even Michelle has data, we didn't show it here, on longer term exposure, because this is exactly what we were kind of saying. And we're looking more than cell death. We're really also looking at other ways, if the cells are not proliferating as fast and are differentiating.
Mm-hmm. Yeah, those are good points.
Thank you very much, everyone. Peter, nice to have you on the call.
Mm-hmm. Okay. Thanks. Any other questions? One interesting, the TP184, which is in the clinic, is that a compound that you got commercially, or were you able to just get access to it for testing?
We got it from our collaborators in Harvard.
Okay.
They were testing it, yeah.
Okay. Thank you.
Okay, thank you very much.
Thank you very much, yeah. Before you go on.
Yes.
Yeah, so the data in the BT869 cell line really captured, my attention quite a bit. So what's Nada's view? What's your view on that particular cell line? Because to me, that's the real cell line for determining real ALK2 biology, in vivo. I don't know if you feel the same way. Because when you look at the survival curve for a lot of the other ones, you just get a couple of days extension. We were looking for model where...
So the BT869, I've been burned before, so we're repeating the experiments just to make sure that we're seeing in terms of... And the other thing that we're doing is we're sacrificing the mice to see if there is any residual. So we had in our Nature Communication paper on when we CRISPR'd out the K27M mutation in another cell line, in BT245. That's another cell, that's not ACVR1 mutant. What we did is we killed the mice. About a year they were fine. And actually we're finding more than 10% human DNA in the brain of the mice. So in a way we've humanized the brain mice just by removing the mutation. This is what I see, why I speak about differentiation in situ. So I don't know if this is what's happening, and I really don't know if this is what's happening when you remove the ACVR1 in any shape and form, repeating the experiment, because I don't trust one experiment. How many mice did you inject for the BT869?
10 knockout clones and around also 8 or 10 parental.
So it's quite significant numbers, but still, we're going to try and find another clone and inject it. I don't believe in clone behavior, so it's interesting. It's going the right way, but I'd rather be very cautious about it. And the rationale behind the sub-Q experiment makes two things. For the H3.1 cell line that are 7M, some of them, a lot of them, don't grow in the brain, like really don't grow. They're the only one that I'm able to grow, and again, maybe Chris and Diana have different experience with that, and I would love to learn from them, but the only one that grows is the DIPG36, and this cell, it's the others don't grow in the brain, so we thought that, let's do it in the sub-Q experiment. In a way, it's even easier to test without having to worry initially about the blood-brain barrier. Test, first of all, are they heterogenic, what's the brain, and then can we treat them, and then after that, go after the brain. Once really have a sturdy model and kind of you think that your drug is hitting its target.
Yeah, there was a question here. Thanks, Nada. There was a question here. They wanted to know what's the length of your assays, and are they 2D or 3D assays?
Sorry, is it you're asking about the in vitro assays?
Yes.
Okay, so for the in vitro assays, I keep the drugs 5 to 7 days.
5 to 7 days?
Kinetic regulators, the AZH2 inhibitors, they require 5 to 7 days, yes.
Yeah, and the question was it 2D or 3D?
No, it's a 2D. It's a 2D.
2D assays.
So we have a 3D organoid model that we haven't yet tested, and I think the moment you start working with this, it's as good to work sub-Q.
Okay. And the other question was how did you determine synergism? What methodology did you use to determine synergism?
I used the CompuSyn program.
Okay. CalcuSyn?
CompuSyn.
CompuSyn, okay.
And if you have any suggestion to us, I'm happy. Again, this is like a new field for us, drug testing, and we didn't embark on it lightly. This is why I started with a genetic model, but then you use it as a tool.
Okay. Yeah, all right. Thank you very much. Any other questions? If not, we'll move on to the next presentation. Thanks, Michelle and Nada.
Yes,
really appreciate that update. Yeah.
Thank you. Bye.
Okay.
Bye-bye.
Bye. How do we go back now? Here?
Yeah.
This one or this one?
The green one, I believe.
This one?
Yes. There you go, yeah, and then select what you want to share.
Oh, you're back in swing again?
That'll be the far left. There you go. That was where you were.
Okay. So we'll continue. So that was a nice little interim, getting some in vivo update in terms of some of the models. So I'm actually close to the end of my presentation. So we were talking about triaging selection of the next round of compounds by the efficacy, or we do some tox or dog telemetry and stuff like that. So those are things we keep discussing over and over again, and I'll say it's a matter of funding strategically, but I think I heard some very good suggestions about looking at some of the ion channels. That's a way to see if you can help mitigate some of the herd liability, if you know what are the ion channels these compounds are hitting.
Do you think it's worth looking at cardiomyocytes as well?
Yeah, that's also very important in a study to also cardiomyocytes in vitro.
Yeah, in vitro.
Yeah, that's a good way to also. I mean, we do the ion channels, we go to cardiomyocytes. I'm always a big fan of the in vivo. Just if we could do the dog telemetry. No matter what in vitro stuff we do, we still have to go. Hello,
This is Peter again. I'm sorry. Since you mentioned the dog telemetry, I mean, one of the things that that we used to do in another organization I was as part of the workup is if there was a positive hERG signal, and I think some of the ones in the upper row of compounds, you know, are certainly ones to consider. That isolated rabbit wedge test be done to look at cardiac channel activity, and you know that can be done more easily and definitely more cheaply than right, the dog telemetry.
Yes, we've talked about that. Yeah, that's a good one too. It's cheaper.
Okay, sorry.
Yeah.
But we'll still take that offline.
Yeah.
But yeah.
That's good points. Okay. So let's speed things up. Oops. Let me just go to full screen here. Okay, so what is it not advancing? Okay, oops. So I thought this was a good point to introduce. So yeah, so there was a recent paper from, Michel Monger from Stanford, and I thought they've actually looked at some studies of combination, which is everybody's thinking a disease like DIPG definitely might require combination in the long run. So it is a very extensive and comprehensive combination screen. And it's interesting, some of the outcomes from this data set were aligning with some of the observations that we've also seen before in terms of HDAC inhibitors. I mean, seems to be very active in these DIPG lines, but in the past they've actually chronically suffered from CNS penetration issues. What's intriguing is the marizumab data, which is a newer compound that's in the clinic by Celgene and Chi-Phase. And they have this proteasome inhibitor. It's one of the most penetrant proteasome inhibitor that's out there, and apparently that compound was also very active across the cell lines. And so they realized there's a nice combination, synergy with the HDAC inhibitor and the proteasome inhibitor, and it's a whole host of them. So it looks like it's a very consistent and robust mechanism. Okay, but like as I said, the issue with a lot of the HDAC is a lot of them are not CNS penetrant. And I've just put some of the combination data below. It looks quite intriguing. You could really see the potentiation or the combination synergism in some of the cell lines. DIPG21, which you told is one of the most sensitive lines that we observe with alpha-biology, you could see shows very, very good synergism, with the HDAC and the proteasome inhibitor. So this is a nice compilation of some information that might be able to use to rationalize going forward. So, I just thinking a lot about the way they have ended up developing, HIV cocktails and stuff eventually for therapy. I actually just put something out there to join. I call it the emerging cocktail for all DIPG. So, like as I said, the problem with the HDAC is that they lack CNS penetration, and so we have been looking for CNS penetrant HDACs, and we have tested a couple of them. So the team have seen, 11-2-1, it's a known class 1 HDAC inhibitor that have gone through some preclinical PET, so we know it actually is very CNS penetrant. So that's a good tool we looked at, and we have actually done some studies in some of the in vitro cell line. But another compound that was really intriguing for us is this FRM0334. That compound, we're actually looking at the CNS penetration in-house, but literature had reported that this is one of the most CNS penetrant HDAC inhibitors out there. It went into the clinic by this company for Alzheimer's disease, and it actually failed in Alzheimer's for lack of efficacy. But nobody have really actually eyed it for a disease like DIPG. So we're trying to gather some data. It has gone to phase 1, and I think phase 1, phase 2. So we're kind of excited about this compound. So in support of some of the data that Michelle just published, if we can now start showing some synergy between these types of compounds and the proteasome inhibitor that's CNS penetrant, and on top of that, all these contribute to the backbone of the disease. We add the underlying mutations of ALK. This could be the triple cocktail that, the disease area was waiting for years. And so,
how selective is the FRM0334 as an HDAC?
It's class 1. I think it hits just the class.
It really does? Very, very specific?
Yeah, class 1 specific. Yeah.
Will not die.
Yeah. All right, so, I think this is a nice pool of candidates to now, evaluate, and we can kind of discuss this a little bit more, but I might get other members of the team to kind of chime in. But it'd be nice to look at first maybe some combinations first, and then we can also consider screening, which of looking at some of the triple. But the nice thing is they're all CNS penetrant compounds, and I think that's one of the Achilles heel for a lot of compounds going after targets like DIPG, so something to think about. Okay, and then lastly, I had Alan.
Can I say something?
Yeah.
I'd like the thoughts of some of the oncologists here, but imagine if we were sequencing melanomas and found a point mutation in a kinase and said, screw that, let's put on some DNA intercalating agents and some HDAC inhibitors because it does have a precise mutation, but let's throw a neutron bomb at the tumors. Why in the world are people thinking that the best route to therapy in a disease where we have two precise mutations, as it were, is to go back 10 years and take some really crappy non-specific thing and blow the cells open just because it kills cells? I mean, and similarly, the other point I want to make is, as much as we can look at some Nada's data and look cool, just as I was advising caution for experiments in Angel's model that don't work, we should not get too excited about looking for a in vitro model that suits our purpose. The ultimate experiment is to get this molecule into children. It has a point mutation in this gene that activates the kinase. We should go as fast as we can and to get clinical data, in my opinion. I'm going back on mute.
Yes. No, we agree. I mean, there's still a path for the development of the ALK inhibitors. Nobody's, against that. I mean, we're still on track. It's not going to affect the development of, an ALK inhibitor for the clinic. But like I said, everybody is now kind of coming to the notion that eventually a disease like this is going to need combination. So we're kind of just entertaining some of those thoughts preclinically, but no, we're not slowing down going after a selective ALK inhibitor for therapy. No.
What's the evidence that everybody says we need combinations? It could make sense, but there's no evidence for that. It's just people think it. I mean, no one's ever put a molecule, so I believe it, but remember, evidence is not what people think.
No, I mean, there's no evidence of absence is a bit hard to get, but I mean, there's nothing has worked so far as a monotherapy, and even like there's ongoing trials combining up to 4 agents, and maybe it's not the right agent, but it's also not working very well. So, yeah.
Yeah, plus too, the underlying nature of the disease is complicated. It's HDAC, it's ALK2, it's PDGF. I mean, there's a whole bunch of underlying, it's not that one clean like FOP like we wish we had. So, people are entertaining that combination might be a way forward. But as I say, it's not going to slow down our strategy in terms of exploring ALK2 as a strategy in the clinic. I agree with you. Yeah. Okay. And then the last slide I had was just to kind of, again, just highlight some of the competitive landscape for ALK2 inhibition, on the clinical front. So far, I mean, other than the Tolera compound, which was acquired by Sunimoto, T184 was touted as an ALK2 inhibitor in the clinic, where you could see, you got a good sense, and you could see from some of the data to do that. It's not a very selective compound, whether it's biology from ALK2 or something else. But all of the other ALK2 inhibitors are in the clinic for FOP, so this is just a highlight of some of the more advanced clinical compound. Thanks to Alex went to an FOP meeting and had provided some of this information and feedback to us, so we really appreciated that. Yeah, but we continue to plug ahead and trying to understand the role of ALK2 in DIPG and trying to come up with a compound that we can aggressively advance to development. Okay, and so to finish up with regards to some of the next steps, like I said, we'd like to complete the scale-up of 2163 and 2117. And like I said, lagging behind those are going to be the 2 amides. We want to continue to look at the backup compounds from CRL and see if we can continue to find something with improved BBB and identify some of these ALK2 protacs which could help us, decipher some of the biology, or also they might be CNS penetrant and be considered for therapy. Key again was some of these mouse models that are under development. I mean, the question was, according to the feedback from the meeting we just had, they gave us enough time to go back and repeat efficacy studies with a new vehicle. That's less than promising. One of the questions we have to ask ourselves, if we're going to do another in vivo efficacy, do we go with another compound or we just repeat 2009? And then we've got some good guidance from NADA. The data from NADA on BT869 was looking interesting, but she wants to validate that. But at least we're getting some preclinical models that we might be able to use to understand if we can demonstrate efficacy. The other point was, again, so we're looking for funding to kind of help triage the next set of compounds. So Owen and I started to explore NIH as an avenue to help support clinical development through a program called Therapeutics for Rare and Neglected Disease. So they provide grants to accelerate preclinical development, and they'll support toxicity evaluation and some of the dark telemetry or the alternative model that Peter just mentioned. So those are ways forward to enable selection of a candidate, and we're hoping to do that within the next 12 to 18 months. And we continue to understand some of these evolving patient-derived lines, to see if we can better understand some of the underlying ALK2 biology. Okay.
So I know I hate bringing this up again, but just kind of related to what Alan was saying, are we viewing the in vivo data as critical path, or is it nice to have? I mean, and I guess if it is, I could see how it would be really nice to have it, and we should do it, but I'm just wondering, is it actually necessary to move into the clinic, or are we putting it as a kind of...
I ask the clinicians what they think.
Yeah.
Right, like if they need that to go in a clinical trial, basically.
Because I feel like that answer should come sooner rather than later, so that we can stage all of our activities.
We had 2 clinical advisory board meetings, which was quite an interesting experience. In so much as there was one group that didn't think it was, really did not, and felt very strongly that there were large groups of clinical sites that would be very happy to proceed directly and move without that. There was another meeting where they were just as strong in the other direction, with the view that this now is moving, that docs need more support for this, and going forward from there. And that is the direction that this will go, because if you have if you have a patient who obviously is a sick child, and you have to make a selection between, let me back up this way. If this is the only drug out there when we go to the clinical trial, I don't think we're going to need a lot of additional support, because there is nothing out there. If there are 2 compounds that a doc or us or anyone is looking at, saying, here are a couple of choices for this, one which has some animal in vivo studies supporting it, you could argue all you want about how valid those are, and one that doesn't but has other stuff. The docs they have to make a selection about what trial they want to put their patient into. And they may go into this based upon there is added animal evidence, and that therefore I'm giving my patient the best chance of survival. And then, of course, there's the regulatory bodies that we have to, we're trying to get discussions started with as well, about how much are they going to require to do this. So this is a slightly long-winded version of saying, we're going to keep going, keep discussing. Peter, one of the nice things that's on the line, Peter Hope, who has been involved from the clinical trial side as well, and he has some opinions and thoughts and meetings with the FDA. Peter, I don't know if you want to jump in on your thoughts for this as well.
Yeah, thank you. I'm not surprised that you've got diverging views from your clinical advisory board. But I guess I'd also, ask you to think that, it ultimately is not, strictly up to the treating clinician, right, as to whether a trial goes forward or not. There are regulatory bodies like Health Canada, like FDA, but there are also institutional review boards, right, IRBs and ethics committees, who tend to be not necessarily, the most progressive, folks, as might be a clinician who has the patient in front of them. And so, they're for better or for worse, and I absolutely understand all the limitations of in vivo models, but that's what they're used to seeing for a cancer drug. And so I think this is something that, should be considered, even if ultimately the data are negative, and there are good reasons for there to be negative because of limitations in the model, at least to show. I do think that one carries, a bit of baggage with any kind of regulatory filing, and I don't mean to the actual regulators, but more to ethics committees and IRBs, to just simply not have any data and say, well, we think it's good enough to go in without. There are going to be some folks who agree with that, but I suspect many wouldn't.
I mean, that's exactly one of the advice we took. So in the beginning and stuff, we kind of wanted to get a feel for whether or not, FOP was enough. And so we met with different clinical groups. And, for disease like DIPG, where the patient pool is not huge, a lot of these clinicians that we met with, some of them are heading up, like, some of these pin-up trials, where the most pool of the patients are. And some of them are very adamant that they're not going to do things the old way, where you can just come and give the kids anything. I mean, they did like our approach in terms of a mechanism-driven approach, and so that actually has legs. I mean, in the old days, when you just take everything toxic, like Al said, and just try it like a Newton from the patients, they actually hate that approach, and they've been getting more and more stringent. So, that can vary from bodies to bodies, but, we put our feelers out there just to get a sense, and we got differences of opinion. But it's something, some of them actually want to see preclinical efficacy model that's related to DIPG, and so that's why we're kind of keeping our options open, not kind of close the doors on any strategy. It's not an easy road to navigate.
Right. So I think the answer to your question is, do we have to do this? And I think the answer to the question is we probably do. We can have, and then we're planning that that's what we're going to do. If the data and if the feedback and everything else tells us that we don't, then we'll change that.
You're basically planning for the worst case scenario, which may be the actual scenario. The worst case being they need this data.
They need this data, and that is what it is. And we can all have theoretical arguments about why it should be or shouldn't be, et cetera, and very valid arguments. But at the end of the day, as Peter was saying, there are many different parties involved here who are used to seeing things in a certain way.
All right, thanks,
And I think having the data would be enabling anyway. Having it doesn't hurt. A clinical word is not going to, if you don't have it, then they'll ask questions. They might, or they may not. But having it probably is not a negative.
Al's hand is up again.
This would be a fascinating time to start involving patient groups and tell the moms and dads, hold it, it's a safe molecule. We have to wait a year though, because we have to make sure it's nice.
Go ahead, I'm sorry. Hello?
You heard me, right, or no?
Your comment was that this would be a good time to get the patient groups involved as well.
Yeah, and explain to the parents who have a child sick that we have to wait a year, even though the molecule is safe and on target, because we want to make sure we can cure some mice first.
Yeah, no, I understand your frustration, Al.
Okay. So should we move on?
Yes.
So Alex, Jungfu, and Alison, you're up. Okay.
I'm going to pass over to Ellie for the first slides, and then we've got Alison and Jungfu. So Ellie, next slide.
Oops, sorry. Oops, too fast.
Okay, so I just want to give you a quick summary of where we are up to on all the crystal structures that we've got. So we've got 4 deposited in the PDB, open to everyone now, fully released in there. You can see the details there. we've got one that I'm currently kind of depositing as we speak, and then we've only got 4 more that need kind of short bits of proofreading and then deposition. So hopefully they'll be done by the end of January, and they'll all be done and dusted, and I'll pass on the relevant sort of PDB codes at that point. You can go to the next slide. So the next thing I just wanted to talk about was some thermal shift analysis that we've recently done, which was screening ALK2 against BMPR1B and TGFBR2. So what we did was we did a TM shift experiment with ALK2 against all the M4K compounds, and I've just divided them up into 6 plates, so labelled 115 to 120, just because that's how it fits in with our internal systems here. But then I also ran the same thermal shift experiments for BMPR1B, and then later I'll show you the TGFBR2 experiments too. And all this shows is how much a compound is stabilizing the protein in question. So in general, a higher shift means more stabilization, which usually equates to tighter binding. So it's a good way of looking at kind of selectivity in a very kind of high throughput method. So I don't know if you can see, but along the bottom here, we have the thermal shift on ALK2. The further along this axis it is, the more stabilizing of ALK2. And then the further up the axis here, the more stabilizing of, in this case, BMPR1B. So anything kind of close to the line, I mean, and the line is fairly arbitrary, but anything kind of on a diagonal isn't really very selective. Whereas anything that's kind of up in this corner or up here would be selective for BMPR1B, and anything kind of down more towards this corner is more selective for ALK2. So here we've got, and I just want to point out that this one is a kind of a false positive, because actually that was a false reading on this particular run, and other data suggests it should sit in the middle of this loop here.
Just as a heads up, I think you're using your pointer, but because we're controlling it, we don't actually see which quarter or quadrants you're from.
Never mind, I'm very sorry. So if things are in the top left corner, so it was working on our screen, sorry about that. So things are in the top left corner of the graph, then they are selective for BMPR1B, and if they're in the bottom right, then they're selective for ALK2. So that's kind of where we're looking for. Anything kind of off the diagonal is going to be the most useful for us in terms of selectivity. So there are a few compounds there that do stand out as being potentially interesting, and I've also marked on all our sort of the 5 lead compounds as well. If we go to the next slide, if you can go to the next slide.
There we go.
Yeah, so this is very, very similar, except this is looking against TGFBR2. And here we can see that actually there's a lot more differentiation. So against BMPR1B, as you might expect, it's quite hard to get selectivity. But against TGFBR2, we are starting to see things that are either quite specific for ALK2, or in some cases, things that are more specific for TGFBR2. So you can kind of see a few of them marked on there. And I'm going to put all this data on the Dropbox as well, and so if anyone wants to kind of look up their favourite M4K compound, then the data will be available, and that's going to be fine. Finally, if you just want to go to the next slide, I've just highlighted a few structures that are selective for kind of the different compounds, different proteins. So anything in the kind of 4-ALK2 box is selective for ALK2, and you can see that we've got kind of fairly common motifs there with the kind of the trimethoxy or the methoxy substitutions down there.
I have to check those because for example, 2-120 is on the bottom right and on the top left. So I wasn't quite sure, but when I was
that might be a mislabeling.
Yeah, when I was labeling, I wasn't sure, but outliers on the graph are in the boxes, but yeah.
So it's quite possible that, yeah, and then obviously in the other corners you've then got the structure elements that might kind of stand out to us as being selective for either BMPR1B or TGFBR2, or some of them were selected for both BMPR1B and TGFBR2 against ALK2. So this might help us kind of piece apart some of the structural elements that are more specific for some proteins than others. Again, this isn't a comprehensive set of structures.
Is there anything here that's helping us prioritize our 5 lead compounds?
So the 2009 is still sticking out as being sort of very selective, which is looking good. The other ones weren't necessarily as selective against BMPR1B. So I think that might help us on that one.
But I don't know BMPR1B is the big major worry at this point.
Yeah. And that's all I've got, and I'm going to hand over to the others now.
Thank you very much.
Thanks,
Okay.
Brian.
Thank you.
So the routine nanobreath screening here, 4 compounds came through from the CRL. Unfortunately, I haven't been able to register the M4K numbers, but hopefully get the data to do that pretty quickly. No real interesting activity in any of those compounds at all, so I won't be progressing those any further, unfortunately.
Next slide. So it's a continuation, like with the theme that a lot of the compound, the epigenetic compound, take a long time to have effect, and it might also be the case for 2009. So this is partly a continuation from our previous meeting, where from Barcelona, the in vivo data, at the end point, when the mouse points were sectioned, it appeared that there was still tumor mass, even with M4K 2009 treatment, and David Jenkinson partly raised that the possibility that 2009 is not very cytotoxic. It might be suppressing growth, but not eliminating the existing tumor mass. So what I did was that I've performed similar viability assay, but instead of just looking at the endpoint where I lyse the cells and measure the basically ATP content in each well, I've also looked at imaged the well day by day based on the staining of permeabilized dead cells by propylium iodide. And after that, I've quantified the proportion of live versus dead cells and then can normalize it against the DMSO control. And indeed, in this experiment, I haven't included the more sensitive line 21, which is also would be interesting to see in subsequent experiments. But over here, I have 007 and number 4, which are having 2 different ALK2 mutations. And I can see that with the 007, it takes up to 4 days to see obvious induction of like cell death. As for DIPG number 4, it takes up to 6 days before I can see obvious cell death. And so I think that the reduction in the viability is probably also partly contributed by the anti-proliferative effect of 2009. But definitely, Ellison is currently doing some experiment, which is acquiring timeless images in an incubator imager to more nicely quantify this with all of our lead compounds. But then she's needing more time to tweak along like the analysis setting as well as the experimental setting. But like hopefully in the future we'll be able to more clearly demonstrate this as well as to dissect the contribution from an induction of, for example, apoptosis as well as like the changes in like Ki67 like staining or like stemness. Yeah. So we look forward to those data.
Okay.
Is there any way that you can extend your assay conditions to look over 14 days instead of 7?
Because if I should be possible, but that would be involving some medium top up or some sort of medium change, but it would be doable as well. But also I'm thinking also what would happen if, for example, for the DIPG number 7, if I withdraw the compound at day 4 or day 5, how fast, like what would happen to the cells, whether they rebound immediately or Yeah, because I think all these results might have some indication on subsequent, like for example, that in vivo dosing schedule, for example.
Okay.
Okay. All right. Okay, thank you. Any questions for Zhongfu? If not, thanks Zhongfu and team for that update.
Thank you.
Let's go on to Lisa. I think it's the last presentation for today.
You've forgotten my one, Methvin.
Oh, sorry. Wow, time is really flying. Yep, you're right. So, sorry about that. Yeah.
No problem.
Yeah.
Okay. I'll go through it quite quickly.
No.
So
Take your time. You have time.
They've been patiently staying there past the end of the day.
I know.
That's all.
a long while till the end of my day. No problem. Yeah, so just generally update, we're continuing to make compounds mainly around the two blueprint patents. As you've already mentioned, we've been making some PROTACs along a strategy of trying to improve the permeability, and I'll talk about that a bit later. So one of the compounds, M4K 1393 in the blueprint series, which we've just recently got the PK data on, and I'll show the data, but there is some improvement. We're not quite where we'd like to be, but we're going in the right direction anyway. I think I mentioned last time that we're working with this AI company, Atomwise, to try and identify compounds with improved selectivity over ALK1 and ALK6. Now, this fits in to some extent with the work that Lisa's doing. So we've completed the synthesis of the first couple of compounds, and they're slowly coming through the synthesis program. And the first couple of compounds went for screening and will be in the next set of results that we receive from RBC. We've been in some discussions with Lisa and Alex and Ellie on the possible fragment screening approach to an alternative possible, quite a very early stage backup series. And I think, you know, we're at the point where we need to have some further sort of offline discussions, and so we're going to try and set some discussions up on that. So I've mentioned synthesis of some PROTACs and some further biology work. I've got a slide on that. And again, I think that's something where we need to discuss the next steps with the guys at the SGC. Yeah, the the turpentine-induced hepcidin model, which Methvin's talked about. And also Julia's making some progress on the evaluation compounds on the patient cell lines, but I haven't got any data on that. If you go on to the next slide, please. So this is just my usual sort of start-off slide. This was the first blueprint compound that we made, which was M4K 3007, which had really nice in vitro ALK2, ALK4, ALK5 profile, but unfortunately it had quite poor. It had relatively high efflux, and when it was evaluated in the PK, it had poor brain penetration. So since then we've been looking to improve the overall physchem properties while maintaining potency and selectivity. So go on to the next slide, please. So I've compared here 3 of the compounds. So there's M4K 3007 in the middle, and I meant to put the data on for the M4K 2256, but the recent compound M4K 3093 has retained reasonably good potency. It has a lower molecular weight than 3007. It also has a lower pKa, and that has now been evaluated in PK, which is on the next slide. So again, I've compared the profiles of the compound that we believe is the blueprint compound 3007 and 3093, and if you look at the table at the bottom, you can see that we have slightly reduced clearance, but it's still relatively high. But compared with the two earlier compounds where the brain to plasma ratio was about 0.1 or 0.2, we're now up closer to 0.5. So there is a significant improvement. Ideally, we'd like to have the brain to plasma ratio up at around 1, but at least we're going in the right direction. Sorry, excuse me. Okay, so these are just some further plans. So based on sort of 3093, we've got these some additional compounds substituted in that same position. We're also going to look at making some compounds with that fluorine with other modifications, where we look at reducing the basicity of that terminal pyrazine nitrogen on the right-hand side of the molecule, and also considering whether we can remove the basic center altogether and replace that pyridyl pyrazine by a pyrazole, as is seen in the second blueprint patent. And at the bottom, we've got a couple of compounds, and so where we've tried to replace the pyrazine on the top by either the amide of an aminopyrrolidine or an aminopyridine, and two of those, the remaining compounds are currently in test. Next slide, please. So this is just reminding you of the second blueprint patent, where they've got a bidentate hinge interaction between the aza indole at the bottom. And we selected 3 compounds. The first 2 compounds, or the first compound with the pyridine, compound 7 was not particularly active. Compound 30 had the pyrazole, and this was the reason we chose the pyrazole for the other series, is a bit more active. Compound 68, while we've been in the meeting, I've just heard that looks like that we now have that completed. We're just in final analysis. So hopefully that will get evaluated in the next assay. And then we can see what we can learn from that patent, and is there anything that we can apply into the other patent. And the next slide, please. So this is just where we're at with the PROTACs. There's a further 3 compounds have been submitted. Again, as I mentioned earlier, we're trying to look at reducing the number of H-bond donors, lowering the TPSA in order to enhance cell permeability and potential brain penetration. At the moment, because we've got chemistry in various other areas, and we've got quite a lot of these PROTACs now, we felt that it was we would stop making new compounds now until we can start to evaluate further in the biology. And I've got just a brief summary update on the next slide, if I don't die before then. So just in summary, in terms of what we've done, we discussed this a little bit at our previous meeting when we were face-to-face in Chester Park. So the only ALK2 antibody that shows any promise is this ABCAM1, and that was the data we discussed last time. The other 2 antibodies, even with high protein concentrations, showed no significant bands. But we do need to do some further validation of that ABCAM antibody with either knockout or knockdown cell lines. We've looked at both the VHL and the CRBN E3 ligases, and they're both present in all of the cell lines that we looked at, except for the 2 mentioned here, the HL60 and the HT1080s, although the HL60 does have the VHL. So that suggests that we should be okay using any of the other cell lines. But it looks from the work that they've done that the HEC293 cells are potentially the most promising, as they express high amounts of both of these. And pending confirmation, the also ALK2. And so this is just sort of a brief snapshot of where we're at. And we will have some further discussions with the guys at the SGC on how to take this forward. And that, I think, is my last slide.
Okay, thank you. Any questions for Sue? Yeah, you made it too.
I've got one question. Sue.
Yeah.
Sue, in this field, is there anything to be learned from in vitro studies with purified, if you had purified cereblon and purified ALK2 protein and mixed it with the PROTACs, looking at how well the two interact, whether by native mass spectrometry or what else, other methods? Is there any lessons from that?
That I don't know the answer to that, I'm afraid.
Okay.
I can discuss with perhaps that's something we can discuss when with my biology colleagues when we.
You can also try for co-crystal structures between ALK2, PROTAC, and VHL, for example.
Yeah, I think there are definitely some other ways that we could look at this. But I'll be in touch, Alex, and we can set a TC up to discuss some further plans and things. Yeah.
Sounds good. Yeah.
Okay.
Okay. Al, you had a question?
Yeah. I see that Nada's lab is still on. Is it possible to send the knockout lines of ALK2 and the wild type control and maybe a heterozygote to Oxford, so that if they've not already, so that we can grow them up, make lysates, and check that ABCAM antibody in the knockout control? And another way to do that, yeah, for sure, just to do that, because ABCAM is very keen on using knockouts as their controls, and Alejandra, who's the vice president of research, would be incredibly keen to support that experiment. But we need the 2 or 3 different lines and the wild type background, or even extract sent to Oxford if it's not already happened.
I will actually speak with Nada about this, and we will get back to you. So it shouldn't be a problem, I think.
Or if you've got them grown up, even better, just make some lysates and send them so we don't have to grow and take the time to grow up the cells.
Okay.
Yeah.
Okay, I will speak with Nada first and get back to you.
Or they could use the same antibody we have here.
Yeah, absolutely. It's just that Olfer's doing the experiment for a bunch of Alzheimer's targets, you know, trying to do it systematically, wild type, knockout, titer, just to make sure that the antibody is of high quality. And maybe there's more than the ABCAM antibody out there. I just think Olfer's gearing up to do these experiments systematically on a whole bunch of antigens, not only this one, and adding this to his quotation marks pipeline would be simpler probably than having someone gear up to do it at McGill or in your group, Alex. So it's just a matter of selling the lines, or even better, the cell, or in addition to the cell lysate, normalized to a certain protein concentration in the lysates. I'll follow up offline with Nada and her team as well, just to get this going, because it'd be super nice to know we have a high quality antibody that definitely recognizes the antigen as is selective. Important for everybody to have those data, I think.
Thanks, Al. This is Jongfu from Oxford. I've actually tested these antibodies in not only in medicine blot, but also in immunofluorescence training to see whether instead of recognizing the denatured protein, it's recognizing the native protein. And among them, the MAB637, actually, I can see like really nice increase in signal with cells overexpressing ALK2. But the thing is, would be nice to validate whether they are clean, if we can have the proper knockout cell line.
Yeah.
Yeah, I would argue, Jungfu, that your experiment is meaningless without the knockout. Just saying.
Yeah. In the same way as we've said, we need to have the knockout to validate this. Okay. Well, if you'll follow up with that, then and we can move forward on that then.
Okay, sounds good. Thank you. Thank you, sir. So we move on to Lisa.
Hello, can you hear me?
Loud and clear.
Excellent.
There you go.
Okay, Okay, so I just wanted to quickly present a little bit of work I did, which is a fragment molecular orbital approach to looking at the interactions between M4K 2009 and ALK2. And this is something that's sort of some experience in this type of approach within the group here. And so it seemed interesting to apply it to the situation where we have M4K 2009, we have a structure bound to ALK2, but we also have M4K 2009 bound to ALK6 as well. So this particular approach explores the interactions between the protein and the ligand by breaking the system up into fragments. So essentially, you're looking at each residue near the binding near the binding pocket, and how each residue interacts with the ligand. And this is kind of you get a sort of a sum of I guess 5 different contributions. You've got sort of charged type interactions in an electrostatic and a charge transfer. You've got sort of repulsion between molecules which are too close. This is the exchange repulsion. The dispersion tends to be sort of hydrophobic type interactions, and the particular version of the program I was using also considers desolvation as well. So I guess looking at this, I wanted to see whether we saw different interaction, or different residues were sort of key in the interactions with ALK2 and ALK6, which might sort of give an idea of how to move forward with selectivity. I know ALK6 isn't necessarily the main concern, but just because they have the same structure bound to both. I've taken this data, I think, from a talk a very long time ago. I don't actually have this data in the database for M4K 2009, but I think I used it in my original project proposal. I said that ALK2 was 13 nanomolar and ALK6 was 68. So you have slight selectivity of this compound for ALK2 over ALK6. I don't know if that fits with the data Ellie actually just presented earlier either. So I'm not sure that it necessarily is different interactions that we're seeing with both, but I'll take you through what was seen and just some thoughts on that. So if you go to the next slide. So this is the output that you get. So at the top, it's the sort of the sum of all the contributions from the 5 different factors. And kind of what you have on the sort of the far right-hand side at the top is things, residues which are contributing a lot to the binding with the ligand. In the middle, I guess, don't need to sort of consider them so much. And on the left-hand side, you would have things which are sort of impacting on the binding, not in a good way. And I was a little bit surprised at the output because I guess the ones that were mainly contributing were not the ones that I was probably expecting. I guess when we look at a crystal structure, and particularly this compound, you see sort of hydrogen bonding with the hinge region, you see some water-mediated hydrogen bonds from the methoxy groups and things, and this kind of just threw a different sort of response, different output than what I was probably expecting we would get. So if you go to the next slide, this will show sort of the M4K 2009 bound to ALK2. I have sort of included in this particular one all the residues that I guess got considered in the calculations. And it was actually the aspartic acid 293 and glutamic acid 287, which kind of sit, I guess, towards the solvent and near the piperazine of the compound, which were showing the strongest interactions. And then the tyrosine 285, kind of at the back of the pocket near the sort of more aryl rings of 2009. And then the glutamic acid 248, the hydrophobic leucine 343 at the bottom. All these residues that I've highlighted here in green, these are actually the same. These are conserved across all the ALKs, so it doesn't necessarily show up anything necessarily for selectivity at this point, but does sort of show perhaps a contribution of the group emerging into the solvent. It's not just sort of solubilizing, it is actually forming some interactions as well. If you take that, I mean, it is a basic amine in this case. If you don't have a basic amine, but you have a group there, it doesn't necessarily lose potency. It might lose a bit, but not totally. So it may not necessarily be a basic amine that's required there, but it appears as though there are some interactions occurring with those residues. Now, if we move on to the next slide, this is the output then from 2009 bound to ALK6. And I've sort of highlighted there were sort of three main differences. And these three differences are actually residues that just weren't included in the input for the ALK2 structure because they just weren't close enough to the ligand. And this is just a different conformation of the two proteins. So having a contribution to binding, the tyrosine 215, this is tyrosine 215 and ALK2 as well, it just sits far away. In this case, it sits very close to the ligand. But what was noticeable was a couple of other residues, the arginine 214 and the lysine 208. So you've got kind of positively charged residues, which are now sort of contributing unfavorably to binding, because that'll be sitting sort of near a charged basic amine of the ligand. So if you go to the next slide, that sort of, now I've trimmed away, this is a very sort of, because it's a very sort of closed pocket in this structure, I had to sort of trim away a lot of the residues. So I've just kept it to those ones that I've mentioned. So in this case, again, you've got the aspartic acid and the glutamic acid towards the solvent and the piperazine, which is showing sort of a high contribution. But also near there, you've got the lysine and the arginine, which are sort of showing an unfavorable contribution to the binding. Whether this in any way gives that very slight selectivity that's seen with 2009 or not is it's hard to say. And it doesn't necessarily point to anything that we can necessarily do with this series. Also, I guess the blueprint, the main blueprint series has a basic amine at this point as well. So it doesn't necessarily guide us with anything to do with these compounds at the moment. But what I did find, just sort of considering it this way, was that perhaps there was some things that we could do to another series, the quinazolone series that was worked on previously here at the ICR. And perhaps there are some things to consider for considering that as a backup perhaps in the future. So I was just going to go through some ideas with regards to what the next steps with quinazolones would be. I guess I'm just putting the ideas out there. Whether there's any chemistry resource to do any of them or not is another matter. But if I just actually, before I take you through that, I suppose I should say, if there's any interest in looking at this type of calculation, for example, on one of the blueprint compounds, we've got the structure with 3007, if that would be in any way useful, it's not particularly it doesn't take a lot of computational time to run this approach. If we go to the next slide, just to sort of present these quinazolone series. So this was worked on here at the ICR. There was a PhD student, Liam Hudson, who worked with Sven Holder, and this work was published last year. And there were sort of two, well, there were these quinazolones. I've taken the M4K IDs, but the data I've taken the data from the publication because I don't have any sort of data to match what we have on our other compounds. So neither of these are in any way as potent as our two main series. And certainly for the smaller compound, the M4K 1097, not particularly selective as well. But both of these crystal structures, Alice Grubers have crystal structures of both of these bound, which show a very different sort of binding mode. So the one on the left-hand side, which has the group coming off the nitrogen, this binds through sort of the bottom nitrogen to the hinge region, whereas the smaller compound sort of flips over and is able to interact with the hinge region through both the oxygen and the NH that is free there. So most of the things I was going to propose actually focus on this smaller compound because it is quite small, and I think there is some opportunities to improve upon the compound. And I guess, you know, from what I saw with the FMO approach, you know, there is extra interactions to be gained as you extend out towards the solvent. So if you go on the next slide, I was proposing sort of a couple of ideas. I've shown these two compounds where you extend out from two different positions on the quinazolinome. I've put a basic amine in here, and I've docked these into ALK2. So this is just to suggest that could we gain extra interactions. Still, the molecular weight still stays quite low, which would be favourable for brain penetration. But obviously, this would need to pick up some extra interactions to improve the potency. The other main issue with this series was selectivity. So I had a couple of thoughts with regards to that. So I've been looking a lot at the compounds we already have. Within the pyridine series, you have the selectivity against ALK4 and ALK5, not necessarily so much against ALK6. The blueprint claim their compound is quite selective against all of those. So I had been sort of trying to work out what drives selectivity against the different ALKs. And if you go on to the next slide, I have my first thoughts on how you could modify this particular series. It's at the moment very potent against ALK6, and so if you take the two ALK2 crystal structures with these quinazolinones and you overlay them with ALK6, you know, these compounds look like they're a great fit within that structure. However, if you were to substitute on the quinoline and sort of extend out that way, that is tolerated in ALK2. You can dock these compounds into ALK2, there's space there. But if you then overlay with ALK6, which I'm showing here, there's a clash with that tyrosine residue, which sits in a different place. So I think there's some rationale for extending at this position, which could gain some selectivity. And lastly, ALK5 selectivity on the next slide, which is a little bit harder to work out with these. In previous compounds with sort of 4 and 5 quinoline substitutions, Alex and Eleanor have already published on their analysis of different sort of hydrogen bonding patterns between the ALK2 and the ALK5 with these 4 and 5 quinolines. I tried to do a similar sort of analysis with two compounds from this series. So this is the M4K 1097. There's also a crystal structure of a very similar compound where that nitrogen in the quinoline is in a bit of a different position. Overlaying those two structures, the water network between the two of them is actually quite similar, and though different to ALK5. So this is going to get, yes, this gets possibly not so clear here. So we've got non-selective 1097, which is the one in pink, and the selective compound 1092, which I've shown in green. The red in that structure is the water from the ALK2 structures, and I've shown a blue for the water in the ALK5 structure. Now you can see the position of the nitrogens a little bit different, and you can also sort of see that that shifts the rings a bit. So whether it's something to do with the positioning of the nitrogen and how it can bond to water in ALK5, or whether if it bonds to, bonds to the water where the rest of that ring sort of sits. I'm not quite sure what would be the most, it's not quite clear for this particular ones what would be necessarily driving that selectivity. Because certainly when you overlay with one structure of ALK5, you see a clash with the aspartic acid 351. But sort of considering what there is there, I think there is opportunities to try and get selectivity for ALK2 over ALK5. You could just go more with the 5 quinoline. It doesn't look too much loss in ALK2 activity. It's slightly less active. Or you could try extending out towards that aspartic acid 351 with sort of a hydrophobic group to see whether that could also have an effect. So yeah, I realize not any current work on this quinazolone series, but this is sort of some ideas that are formulated. So if there's any interest at all in following up on this, if there is any chemistry resource, do let me know.
Okay, that's it.
Yep.
Yep, thank you. Thank you for that nice analysis. All right, any other questions from Lisa? Okay. If not, thanks everyone for excellent meeting, discussions.
If you want to take a second to talk about January and what we're doing and the timeframes and go from there.
Yep, so this is the last meeting for the year. Usually people come back at different times in January. We actually don't have a meeting in January because we typically have the meetings in the early part of the month. But some people are just coming back. So our next meeting is in February, I think February 12th. Yeah. So hopefully we get enough time to gather some more data and address some of the burning questions and issues, and looking forward to some more updates later.
Yeah, and in the interim between now and then in January, I think we're going to try and schedule a couple one-on-one smaller group meetings with both Charles River, Sarah talked about some of that. Also, we're going to have a planning session, which I'm going to try and set up for a smaller group and make a decision about which studies we're going to move forward between now and the end of March. And so we'll go from there. So though we won't be having a meeting due to the holidays, we will be trying to have a couple other conversations to keep the critical path moving forward.
Beautiful. Yeah.
All right.
That's a good point. Great. Thank you. Thanks everyone.
Thank you.
Enjoy the holiday.
Yeah, have a great holiday everyone. Thanks for joining.
Don't drink too much.
That's the whole point of the holiday.
All right, take care.
Bye.
Thanks everyone.
Bye.
Bye.
Bye.